to the chat box for your questions so that we have them all consolidated into one place. Thank you. And now I'm going to pass things off to Dr. Keith Alcohon.
Okay, so pleasure to introduce Jonathan Himmelfarb. Dr. Himmelfarb is the holder of the Untitled Chapter for Kidney Research in the Division of Nephrology. He's also director of the Kidney Research Institute. He's nationally and internationally recognized leader in nephrology. He's been a past president of the American Society of Nephrology and is a standing member of the NIH CDK study section. With regard to his research contributions, his early publications focused on dialysis, dealing with metabolic complications associated with uremia and dialysis-associated inflammation, oxidative stress, insulin resistance. And he's also got a number of high-profile publications and clinical trial consortia. Most recently, Dr. Himmelfarb and collaborators have been involved in developing a micro-physiological system to study certain aspects of kidney function in vitro. This so-called kidney on a chip platform is proving to give unique insights into kidney disease pathogenesis. In fact, this system caught our attention and looks like a very promising way to investigate several aspects of lupus nephritis. So members of our division are working with Dr. Himmelfarb on the role of neutrophil nets and serological components in a recently funded DoD grant application that looks to be very promising. So we're very happy to have you here, Jonathan, and look forward to your lecture.
Okay. Thanks very much, Keith, and thanks, Lara. So the topic is kidney precision medicine for this talk. These are disclosures. And what I'll try to cover in the next 45, 50 minutes, what do we mean by precision medicine? Why focus on the kidney when we think about precision medicine? Keith mentioned some of our work developing new in vitro systems for clinical trials in chips or in a dish. And I'll also focus on some of our work with stem cells and human kidney organoids, and then also a large project called the Kidney Precision Medicine Project that's funded by the NIH. So I'll start with this slide from Margaret Hamburg. This is a decade ago when at the time, she was the commissioner of the FDA, and in the New England Journal of Medicine, she used strong language. She said, "We are failing as a scientific community and as a nation to produce the science, the promise of science, to use that to deliver treatments, preventions, and cures for diseases." So she made a call for new methods, assays, standards, and models that'll help speed the development, review, and approval of medical products that patients need and rely on. So one of the leaders in American medicine using strong language in the New England Journal saying, "We are failing." And Francis Collins used similar, as the director of the NIH, very similar strong language in saying drug development in the United States is in crisis. A whole series of publications where he talked about that we think we have an understanding of the molecular basis of about 5,700 human diseases, but we have safe and effective treatments for less than 10%. And it costs several $billion. And from the time somebody at the bench comes up with a discovery that's eventually going to lead to a new therapeutic, it takes on average between 14 and 17 years before patients see that, before it becomes a new drug. And despite all the preclinical work that we do, 70 to 90% of drugs, new drugs, fail in clinical trials, and a small percentage are proven ultimately to be safe and effective. And a lot of questions have been raised about the relevance and the reproducibility of animal models. And so Francis Collins and Harold Varmus, another former NIH director and Nobel laureate, wrote in the New England Journal Perspective on Precision Medicine about 5 years ago now. And this is really their concept, that it's not one size fits all. It's not finding the blockbuster treatment anymore that you can give to everybody with a given condition and it's going to make a big difference. It's more like finding the right drug or the right treatment at the right dose at the right time for the right patient. So it's really, instead of lumping everybody together in groups, as we've often done for the last 20 years or so, starting to, again, individualize our thinking about disease when we talk about precision medicine. This is the precision medicine. This is sort of a conceptual figure of the precision medicine clinical trials concept that is really behind what Francis Collins is talking about. So people are quite heterogeneous here in different colors here. And let's say we want to make a treatment for any medical condition. Let's say for the moment we're talking about a heart attack. We have a drug that we think is going to prevent heart attacks. Well, in this heterogeneous group of people, let's say we want to enroll this group of people in a clinical trial. There are people who are destined to have a heart attack, here in blue and yellow, to have an event. And in that group of people, there probably half the group in this case was destined never to have a heart attack, the people in green and the people in red. So if we have a drug that prevents heart attacks and we give it to this group of people right here, there's going to be no effect of the drug because they weren't going to have a heart attack anyways. And amongst those who are destined to have a heart attack, our drug may be targeting a pathway for which a group is responsive here in blue and a group here in yellow would not respond to that particular pathway because that's not what's driving the disease pathogenesis in those individuals. So if we do a clinical trial with this group here, this is the only group that might respond to the treatment. And that's why in these large clinical trials, an effect size of 15 or 20% is considered a good outcome, because really it's a minority of the people in the trial for whom the drug may or may, or the treatment may or may not be effective. And because of that, we need large trials with 10 or 20,000 people in them to move the curve from here to here and get that effect size of 15 or 20%. So the precision medicine clinical trials concept is that if we knew a priori before we started a clinical trial what were the drivers of the disease in the population and who was destined to have an event, and we targeted that population for our clinical trial, we'd come up with more effective treatments and it would be easier to prove they're beneficial and smaller clinical trials and cost less money. So that's the precision medicine clinical trials concept. And Shinya Yamanaka, who won the Nobel Prize for discovering or finding a way to make reprogram cells to make inducible pluripotent stem cells, it's another visionary, and he came up with the idea of in vitro precision clinical trials, that concept. And it's essentially the same concept, but now we take cells from an individual, reprogram them into stem cells, inducible pluripotent stem cells, differentiate them into the tissue or the organ system that we think we want a treatment for, and then explore the differences, in this case largely genetic differences, and find the responders in vitro and the non-responders in vitro, and then go to clinical trials only with the responders for a given treatment. So it's the same concept for the large clinical trials, but it's using in vitro assays precisionally in order to enhance those ultimate clinical trials involving patients. And this is the Economist idea of precision medicine. So the Economist, every December, they just put out an issue this last week. Every December they have an issue where they talk about what they think is going to happen in the following year. And this was their idea of precision medicine. So you have this sort of androgynous individual here. You've got a double helix that's barcoded. You have a pill with the same barcode on it. You've got some kind of drug in here and some kind of petri dish here. This has got the same barcode on it and it says 100% match. So the public's idea of precision medicine is hyped to where the bar for us as a scientific community is quite high if we're going to meet that bar. Now, coming back to kidney diseases, they're a public health problem, and I think everyone is aware of this, but the extent of kidney disease as a public health problem is really quite remarkable, and we learn more all the time. But about 37 million Americans and about 850 million people worldwide have some type of kidney disease, 9th leading cause of death. We've had this rise in kidney failure or end-stage kidney disease. Unfortunately, survival on dialysis is short and quality of life is not good. The kidney diseases, it's actually up to about $120 billion now, estimates are annually. And we have not, as a community, kidney community, developed very many new safe and effective therapies or cures, although I will say the SGLT2 inhibitors are changing the landscape pretty quickly. So one of the reasons, why has it been hard to make good therapies for the kidney? Well, the kidney is a really complex organ. It's got an incredibly complex architecture with these filtering units, the glomerulus, with this intricate structure of these podocytes here that create 140 liters of filtrate a day, work 24/7. Kidney is an incredibly vascular organ. About 20% of the volume of the kidney is blood vessels. These two small fist-sized organs get 20 to 25% of the cardiac output. So you form this from 700 liters a day of blood that enters the kidneys. You form this 140 liter filtrate that then gets processed by this complex tubular network that has at least 40 different cell types in it and is entirely surrounded by a vasculature. And the job of the kidneys is to maintain homeostasis throughout the body by maintaining constancy in the composition of the blood. So architecturally complicated organ, functionally complicated organ, multiple different cell types, really analogous in complexity to the brain. And it's not really a surprise that the kidney and the brain have been two of the organs for which it's been very difficult to develop new therapies over time. So Francis Collins, when he became the director of the NIH, created NCATS, the National Center for Accelerating Translational Science, and their signature program that they put out an RFA, request for applications, for about 8 years ago now was to create human organs on chips for precision medicine. This was the first time ever that the NIH partnered with DARPA. Which is the defense agency that created GPS and the internet, all kinds of things. And the goal was to create 10 different organ system mimetics that would be physiologically relevant, so-called human organs on chips. They could be used for preclinical safety and toxicology for drug screening. They could represent human disease for proof of concept studies, be an alternative to animal model testing. And because most of our animal model research, we use a strain that is genetically identical, they're on the same diet. Usually we try to keep all the parameters as constant as possible. Animal model testing never imitates the heterogeneity of human beings. But these organs on chips could be genetically diverse and they could be conceivably suitable for use in clinical trial settings. So this was back in 2011, 2012. And we competed to develop a kidney on a chip and thought that if there's an organ system for which these micro-physiological systems or humans on chips are relevant that incorporate microfluidics, the kidney should be one of those systems, because with those 700 liters a day of blood flow and that 140 liter filtrate, in the tubules, 99% of which gets reabsorbed. Fluidics is critical, and every cell in the kidney is touched by fluidics. And so for this initial grant proposal, we partnered with a small company. At that time, it had 5 employees, called Nortis. Nortis was spun out of UW Bioengineering, and when we started this, they were in Fluke Hall. It was then called the Center for Commercialization, the Tech Transfer Office of UW. So they were basically a UW-based company, and this was the first chip that we used. And what you see here, in this space here, you can bioengineer a tubular structure. Volume is about 700 nanoliters, so about 1,000th the size of a drop of water. So we're talking about bioengineering tiny structures. You could cross-perfuse, so you could mimic nutrient flow and lymphatic outflow. You could perfuse luminally. These are bubble traps here. So you could put in the media of your choice, and then you can collect all the effluent that comes out would be the equivalent, if we're bioengineering a tubule, of the filtrate that becomes the urine. This whole system is filled with extracellular matrix, so it's highly biocompatible. The cells never see the material of the chip. They only touch matrix as they would in vivo. And these are Nortis now spun out of UW, now has a big plant in Woodinville, and is a bona fide company. And these are the newest generations of their chips. This is a triple channel chip now where we can do 3 experiments on 1 chip. And to start with, we made the strategic decision not to try to emulate the entire complexity of the kidney with its 40 cell types, but because we were interested in drug elimination or drug disposition and drug-induced toxicity for the kidney, we focused on 3 cell types: the proximal tubule, and the peri-tubular microvasculature, and the peri-tubular microvascular endothelial cells, and the perivascular cells, or so-called pericytes. And this is the architecture of healthy, this is histology from healthy human kidney, the cortex. What you see is it's chock full of proximal tubules in green here, and they're completely surrounded by a vasculature, which you see in blue and you see in red here. And these pericytes they really control the microcirculation of the endothelia. So we felt if we could incorporate these cell types into a model, we could understand how the kidney eliminates drugs, and we could understand most drug-induced nephrotoxicity, because the proximal tubule is the target for most nephrotoxicity. And also, I should add, for acute kidney injury. So it took us, after we got funded, a couple of years and almost a dozen people working on this and a lot of trial and error, but we did learn eventually how to reproducibly bioengineer these proximal tubules, human proximal tubules on a chip. And they maintain their phenotype in vitro for weeks or months at a time, very different than a standard 2-dimensional culture where cells undergo epithelial mesenchymal transition and they lose their functionality. And we had, these were similar to DARPA, milestone-driven performance grants, and we had to prove that our proximal tubule on chip functionally looked exactly like a human tubule, and we were able to do that for all the functions of a proximal tubule. Having done that, we wanted to start modeling kidney diseases. And there are a lot of kidney diseases. Obviously, the kidney is a complex organ, and lots of different cell types can be injured. And like many really organ systems that are subject to disease, there are gene-environmental interactions, and there are some kidney diseases that are dominantly genetic, like autosomal dominant or recessive polycystic kidney disease, or we'll come to APOL1. And there are some that have very little genetic component, but are largely environmental, like toxic acute kidney injury or traumatic acute kidney injury, and a lot of diseases in the middle with both a significant genetic and environmental component, like diabetes and hypertension or autoimmune diseases. So the basic construct that we had for this work was to say, there's lots of different cell types in the kidney, they can be injured, and there are probably specific initiators and mediators of that injury, and we can model the injury in vitro, and then see what path the response to injury goes down. Does it go down a reparative pathway, or does it go down a pro-fibrotic, pro-inflammatory pathway that leads to progressive disease, or does it go down a regenerative pathway? So the first thing we wanted to do was establish biomarkers of these different pathways in vitro for these in vitro systems. And we published 3 different papers in our lead journal, the Journal of the American Society of Nephrology. The first one, we identified upregulation of cell-associated heme oxygenase-I in proximal tubules as a very prevalent proximal tubule toxicity marker, drug-induced toxicity marker. We also identified in the effluent kidney injury molecule 1 as a very effective biomarker of proximal tubule injury, very analogous to KIM1, which is now used. It's FDA cleared as a in vivo human kidney toxicity biomarker. Then we developed biomarkers of fibrosis that we could use in these systems as well. And then we also developed biomarkers of regenerative capacity. So armed with those tools, we could now start to model various diseases. And I'll give you one example that shows the power of these kinds of in vitro systems. So there's an entity that we now call Aristolochic acid nephropathy, which has an interesting history. It was discovered about 25 years ago in Europe, where there were 105 young, healthy women who were in a weight loss clinic in Belgium. And these 105 women came down with severe progressive kidney disease leading to kidney failure. And then subsequently, many of the women developed urinary tract malignancies, all within a matter of months. That obviously caught people's attention. And there was a CDC-like investigation, and the common, what was thought to be etiologic agent was that there was a mistake made in an herbal preparation that the women were taking, and Aristolochia or Aristolochic acid was substituted in large quantities for another Chinese herb. And after that discovery, and that was published in the New England Journal and the Lancet back literally about 25 years ago. After that, there had been an entity that had been known for 100 years called Balkan endemic nephropathy, mysterious entity where there are villages along the Danube River in the Balkans that were known for decades. There were high rates of kidney failure. And people figured there was an environmental exposure, but people had looked for all kinds of exposures and hadn't found one that was consistent. But after this description of Aristolochic acid causing Chinese herb nephropathy, people discovered that along the riverbanks in those towns on the Danube where there was a lot of kidney disease, Aristolochia, which is a plant, was growing there. And it was getting the seeds from the Aristolochia were getting mixed up with the wheat that was being grown in those villages. So this was, whereas Chinese herb nephropathy was high dose Aristolochic acid acute exposure, Balkan endemic nephropathy is a chronic low dose exposure. And it's now, Aristolochic acid is now recognized as a class 1 carcinogen. It adducts DNA and adducts proteins, and that's how it's mutagenic and carcinogenic. And it's still estimated that about 100 million people worldwide are exposed to Aristolochic acid. It's banned in the United States, but you can still buy it on the internet. Taiwan, it's used with high frequency in Taiwan in herbal preparations, and Taiwan has the highest rates of kidney failure per capita of any place in the world. So in the 25 years since then, there have been a lot of animal model research, but it hadn't really established, there was a lot of controversy about how Aristolochic acid was nephrotoxic. It had been pretty much assumed that it has to be bioactivated and that probably happens in the liver in order to be nephrotoxic. But again, the pathways of metabolic bioactivation weren't clear. So we had an enterprising graduate student at the time, Shirley Chang, who simply took our kidney on a chip, put human hepatocytes in a chip to make a liver on a chip, and then mixed our kidney media and liver media 1-to-1 and perfused through the liver to the kidney Aristolochic acid. And what she found, and it's, sometimes you only need simple readouts in experiments. So she did live dead staining, where green is live and red is dead. Aristolochic acid, 0, 10, or 25 micromolar. And she either perfused the kidney directly or through the liver. And what you see, you know, with no Aristolochic acid, everything is green, no cell death. With 10 micromolar Aristolochic acid, the kidney looks fine, direct perfusion. But you start to see some necrosis, some dead cells when you pre-perfuse through the liver. And at 25 micromolar, the entire tubule is now necrotic. And even in the kidney, at high concentration, you start to see a little bit of injury. She then got more sophisticated, and we had a polyclonal antibody that detects these Aristolochic acid DNA adducts, and she showed that even at 10 micromolar, liver kidney, when you pre-perfuse through the liver, then all these adducts are forming to the DNA, which you don't see with direct kidney exposure. So she clearly identified that the liver hepatocytes bioactivate aristolochic acid and make it nephrotoxic. The next question was, could we work through these pathways, metabolic pathways, which were controversial in animal model studies? And they're both cytochrome P450 oxidases in the liver, which were thought to be a detoxification pathway, and NQO1 and other reductases that add an N-hydroxyl group into aristolochic acid. And these can then be, there are lots of sulfur transferases in the liver, it can be sulfated. And there was a lot of controversy about which of these was the etiologic agent that causes toxicity. So she reasoned that whatever was the toxic agent, if you perfused it directly into the kidney, it would be just as toxic. You wouldn't need to pre-activate it by hepatocyte exposure. So here she showed the N-hydroxide, you perfuse it directly into the kidney, everything's still green, so there's no toxicity whatsoever. But the sulfated form completely necrosed the tubule with direct kidney perfusion. And she showed quantitatively that it was just as toxic as if it had been perfused through the liver first. And that made a lot of sense to us because the kidney excretes a lot of sulfated compounds via basolateral secretion through organic anionic transporters on the basolateral surface of the proximal tubule. These are also the transporters that eliminate many drugs from the body as well. So sulfated compounds are targeted for proximal tubule elimination, but they'll passage through the proximal tubule to get to the lumen and the filtrate and what becomes the urine. So if that's the case, if this is, if these are entering the proximal tubule via these OATs, organic anion transporters, that you should be able to block it. And probenecid is often used as a probe, a competitive inhibitor of the OATs. And she showed, yes, if you pre-treat it with probenecid, then you could completely block the nephrotoxicity. So she did more work and showed how the liver sulfur transferases form this compound, how they're transported out of the liver. But basically, one graduate student in 3 months of work ended 20 years of controversy about the multi-step mechanism by which aristolochic acid becomes nephrotoxic. So it shows the power of these in vitro systems because you can control all the parameters in ways you can't. In vivo, it's very hard to do this because these reactions are all instantaneous. So I'm going to switch gears now to the vasculature. I mentioned at the very beginning that the kidney is the most vascular organ in the human body with those 700 liters of blood flow a day. And it has a very complex microcirculation as well, where most of the kidney is perfused via the postglomerular venous outflow. The efferent, actually it's arteriolar outflow, but efferent arteriolar outflow. And there's been very little work really carefully examining the human kidney microvasculature. So for this work, we partnered with a biomedical engineer and vascular biologist, Ying Zheng at UW, and she had previously developed these beautiful human microvascular networks that you see here with a system that she developed in her laboratory. And working with Ying on this project, again, it took a couple of years of work, but we were the first group to isolate, learn how to grow and characterize human peri-tubular microvascular endothelial cells. And then we put them into these bioengineered networks. And what you see here on the left, this is histology of the normal human kidney cortex, kind of like we showed at the very beginning. What you see, blue are the nuclei from all these proximal tubules. And you see how every tubule is completely surrounded by a vasculature. And you see that these pericytes, these perivascular cells, are always at the inflection points where they can control the microcirculation. This is our bioengineered human kidney peritubular microvascular network. And you see here again how these perivascular cells are always at the inflection points where they can control the microcirculation. So you can see how close this looks to the in vivo microvasculature, this in vitro system that we have. And this is, again, normal human kidney histology, and these are the glomeruli here. And this is stained for VWF, von Willebrand factor, and something called PV1, which is a plasma-lemmal protein that's involved in the formation of fenestrae. And you see that the kidney glomerular microvasculature is enriched in VWF compared to the peri-tubular microvasculature. And conversely, the peri-tubular microvasculature is enriched in PV1 compared to, there's almost none in the glomerulus. And here you see we've recreated in vitro these microvascular networks, here peri-tubular and here glomerular. And you can see it's remarkable to me that they maintain their phenotype in vitro. So you see a lot of VWF in the glomerular microvascular cells that are grown into a network. And conversely, you see a lot of PV1 in the peri-tubular microvascular networks. They also maintain this fenestrated structure. So the kidney microvasculature, both glomerular and peri-tubular, is highly fenestrated, meaning that it transports a lot of substances. And that makes perfect sense because you're forming that filtrate coming out of the glomerulus. So it has to get across the endothelium, and so a fenestrated endothelium allows transport. And there's so much reabsorption and secretion between the peri-tubular microvasculature and the tubular epithelia. There are differences. The peri-tubular microvasculature has diaphragms in the fenestrae. Developmentally, glomerular microvascular starts with diaphragms, but lose them so that adult glomerular endothelial cells are fenestrated, but without those diaphragms. So having recapitulated the function and the ultrastructure of the human kidney microvasculature, we set out to make a model of cyclosporin thrombotic microangiopathy. And we chose cyclosporin for a couple of reasons. One is it's still a big clinical problem. The second reason is that the preclinical work before cyclosporin was FDA approved did not identify significant nephrotoxicity. And it's still very hard today to make a good animal model of cyclosporin nephrotoxicity. You have to use 10-fold higher doses, salt deplete the animals, possibly give them NSAIDs to get nephrotoxicity. So we thought this is a good test case. Could we make a thrombotic angiopathy model that was kidney specific using kidney endothelial cells? And the answer was yes. That when we take kidney microvascular endothelial cells, but not say human umbilical vein endothelial cells, and expose them to cyclosporin, we could recapitulate a thrombotic microangiopathy where you see labeled platelets here in green. This is the luminal surface of the vessel, and erythrocytes binding to the luminal surface of the vessel, in essence, recreating a thrombotic microangiopathy. And then from a drug discovery perspective, we're interested in inhibiting this. And for drug discovery, you often want to have what are called target engagement biomarkers. So you want to know that you have an easy readout for a biological effect. And we identified as a target engagement biomarker. Basically, this is based on cyclosporin and tacrolimus are calcineurin antagonists. And downstream of calcineurin, NFAT gets dephosphorylated and then translocates into the nucleus. If you give VEGF, vascular endothelial growth factor, to endothelial cells, you see very nicely here that one isoform, NFAT1, of this transcription factor completely translocates into the nucleus. You can see it here inside the nucleus, whereas here you can see there's none of it inside the nucleus. And it turns out cyclosporin completely inhibits that translocation of NFAT1 into the nucleus. So we use that as a target engagement factor. And we worked with another young investigator, Alexis Kashansky, a computational biologist at Seattle Children's, and did what's called used a kinase regression approach to see if we could rescue the vascular injury from cyclosporin. by using 28 kinase inhibitors, and again, a machine learning approach to, because there's a lot of redundancy and promiscuity to these kinase inhibitors, and have come up with some molecular targets for rescuing vascular injury from cyclosporin, and we're now starting to explore the same with Shiga toxin and other causes of thrombotic microangiopathy. So again, a nice example of how you can use these in vitro systems to learn more, to disease model, to learn more about human disease, and potentially come up with therapeutic targets. So I'll switch gears again to another one of our young investigators that work with us, Bino Friedman, who is one of the leaders in national leaders, and actually he's this year's Fialco awardee at the University of Washington as an assistant professor who's doing great research. And Bino, who at the time was in Boston, was one of the first people to figure out how to make human kidney organoids from inducible pluripotent stem cells. And these kidney organoids, here you see sort of the normal human kidney architecture they're small, but they resemble the architecture of the human kidney with glomeruli, podocytes, if you will, in glomerular structures, and then tubular structures that connect to a certain degree to these glomerular structures, but with nowhere near the maturity and the complexity of the human kidney, but they're models for the human kidney. And remarkably now, and this to me is really kind of amazing, but we can take a urine sample. Urine has live cells in it. We can take a urine sample and then de-differentiate those cells and make inducible pluripotent stem cells from those cells, and then grow a kidney organoid. So we can now grow a kidney organoid from a urine sample, and we have a protocol now where people with a variety of different kidney diseases from around the world send us urine samples, which we reprogram into stem cells and can make kidney organoids for basically disease modeling. And Bino has really focused and become the leader in studying polycystic kidney disease, which is one of the most common genetic diseases to have a serious phenotype, and certainly for kidney diseases is by far the most common. It affects between 1 in 600 and 1 in 1,000 people worldwide. It tends to cause kidney failure later in life in people in their 40s, 50s, 60s, 70s. And 25 years ago now, the genes involved were cloned. Polycystin-1 and polycystin-2 are the proteins, and PKD1 and PKD2 are the genes. And literally it was 25 years ago last year that PKD1 was cloned. I remember thinking at the time, this is great. Now that we have the genes and now that we have the proteins, we'll come up with a cure for polycystic kidney disease. But we're 25 years out and we don't have good therapies, and there's a lot of controversy about just how these two proteins cause disease. We do know, just to go back, we do know that this is a orphan G protein coupled receptor, polycystin-1, and this is a cationic channel, and that they do bind together, and they tend to be in the cilia, but how they cause disease is not fully known. So Bino led an effort to use what was then brand new technology, CRISPR-Cas9, back in 2014 or so. And his group was the first to combine CRISPR with inducible pluripotent stem cell derived organoids to create a disease model of polycystic kidney disease. So they basically CRISPR'd out either PKD1 or PKD2 and found that then the organoids actually form cysts. It's quite remarkable in vitro. And then we recruited Bino 6 years ago to UW, and he's continued this work, and Nellie Cruz, working in Bino's lab, came up with a mechanism or a method to increase the cystogenesis rate in these organoids from what was originally reported around 7% up to about 70%. And that's high enough to use for phenotypic screening for drug discovery purposes. And it's quite remarkable. I'll play this movie. You can watch now here. If you watch right here, this is a kidney organoid time-lapse photography over a couple of weeks. You can actually see a cyst form. So biophysically, we can now watch and understand how a cyst forms using human or kidney organoid cells, tubular cells in kidney organoids. That's PKD gene specific, because you don't see these cysts unless you have edited out either PKD1 or PKD2. So it gives us incredible insights into the pathogenesis of cystogenesis, which we never had before. And because we are now at a level where you can screen for phenotypic screening, that's exactly what Bino's lab has done using robotics, mini kidneys, and screened panels of different drugs to prevent cystogenesis. And remarkably, this work has identified a new mechanistic pathway related to myosin and the cytoskeleton that had never previously been suspected to be involved in polycystic kidney disease, even with the 25 years of animal model research that has been done. And then we've taken a compound that we, from that screening, and Louisa Helms, who's a grad student right now, has taken a compound that worked in vitro and now put it into animal models of PKD and found that you can inhibit cystogenesis and you can even regress cysts after they've already formed. So again, discovery that you can make in vitro that you can then bring to clinical trials in vivo. One other disease, so I think that organoids are particularly useful for genetic diseases when you combine them with gene editing technologies. And one disease we're very interested in is understanding why African Americans, particularly of West African origin, develop kidney failure at very high rates. So if you're born African American in the United States, you have about 1 in 12 lifetime incidence of developing kidney failure. Right now, African Americans are about 12 or 13% of the US population, about 35% of the dialysis population. So it's a huge public health crisis or issue that we'd like to be able to do something about. And we've known for a long time that kidney failure in African Americans tends to run in families. And the NIH funded efforts to try to figure out genetic risk amongst African Americans for kidney failure. And Martin Pollack at the Beth Israel 10 years ago published a beautiful paper in Science where he identified APOL1 variants as being highly prevalent in African Americans and strongly associated with risk for progression to kidney failure, irrespective almost of the initiating cause of kidney disease. And he related this in a story that's somewhat similar to the sickle cell story. APOL1 travels on HDL and it's protective against African sleeping sickness or trypanosomiasis. So these mutations develop, particularly in West Africa, became highly prevalent and they're protective against trypanosomiasis, but they're maladaptive for Western type diseases that happen later on in life, like progressive kidney disease. It's been a challenge because this is a late and it's not evolutionarily conserved and it's only in primates, APOL1. And so it's been very difficult to make animal models to try to understand APOL1 mediated kidney disease. It's thought to be a two-hit disease. Something else initiates injury. And then if you have these risk alleles, and 13% of African Americans are homozygous for these risk alleles, then it dramatically increases your risk of developing kidney failure. So working with a young investigator, Jenny Lin at Northwestern, we used CRISPR to knock in the risk variants into kidney organoids, first into inducible pluripotent stem cells and then into kidney organoids. And here you see that knocked in, the mutants that are knocked in, you can grow the organoids and they look perfectly normal. Here, the podocytes, the proximal tubule cells, distal tubule cells, and the organoids, they look normal. So you can make a model. And we've used single cell seq techniques, again, here working with a young investigator at Michigan. we published the first paper using single-cell RNA-seq methodology to study kidney organoids, and then applied that to create a model of APOL1 disease. And we can identify that gamma interferon upregulates APOL1 several hundredfold, particularly in podocytes. And we think that APOL1 disease is normally a proteinuric disease, and that it involves podocytes as the primary target. So we're now in the process, we've created a model in vitro for APOL1 where if you overexpress it with gamma interferon and then you add an ER stress agent, tunicamycin, you can get upregulation of inflammatory cytokines, heat shock proteins, similar to what we think happens in vivo. So we're now actively investigating using these organoids and CRISPR to see if we can come up with a phenotypic screen for APOL1 and drug discovery for mediating APOL1. So in the last few minutes, I'm going to completely switch gears and talk not about our in vitro systems, but what needs to be done in vivo to really advance the public health related to kidney diseases and try to prevent kidney failure. And the 500,000 people in the United States, or 700,000 people in the United States currently have end-stage kidney disease, 500,000 on dialysis, a couple 100,000 that have been transplanted. And we use the eGFR as our primary measure in albuminuria for staging kidney diseases, but we know they're not very precise. And this talk is really about precision medicine. Abole was a European pathologist who back 50 years ago published a whole series of papers showing, he looked at 40,000, he was a nephropathologist, 40,000 biopsies during his career, and he consistently showed there's a really poor relationship between what's going on in the kidney and the GFR. So here you see from people with identical creatinine clearances to glomeruli, one looking totally sclerosed and the other looking 100% normal. And we know that diabetic nephropathy, for example, which is a huge public health problem in this country, what we see clinically is the tip of the iceberg. So this was an interesting paper where at autopsy, diabetics who died, who had been followed carefully and were known not to have albuminuria and to have normal GFR. So these are people in whom clinically we would have said they have diabetes but no kidney disease, and at autopsy they have clear-cut diabetic nephropathy. You see in here mesangial sclerosis, nodules that are characteristic of diabetic nephropathy. So what we see clinically is the tip of the iceberg for what's going on actually in the kidneys. And generally speaking, people with diabetes and hypertension and progressive kidney disease don't get kidney biopsied clinically. They just get treated with RAS inhibitors and now probably SGLT2 inhibitors. And if they progress, and we don't do anything about it. So the NIH decided, the NIDDK, that this was the biggest problem, the biggest public health problem to which they could make a contribution. And so they created what's called the Kidney Precision Medicine Project. And the goal of this was to obtain kidney biopsies from people with either acute kidney injury or chronic kidney disease due to hypertension or diabetes for research purposes. And to use these biopsies to create a kidney tissue atlas and identify cells, pathways, and targets for therapies, stratify patients, and again, from a precision medicine perspective, develop individualized treatments. So just because you have diabetes and you have some degree of kidney dysfunction doesn't mean that you're all the same. And the pathways that are active in the kidneys are likely to be highly different, that are driving the disease process among different individuals. So the goal here was to create an atlas, a so-called atlas that would go into the public domain. And the idea of these atlases, there are now a lot of efforts to create different tissue atlases through the NIH and through other organizations. And the idea is not so much like here was the first atlas, or even when I was a kid, you know, if you wanted to drive from one place to another, you opened up the Rand McNally Atlas and, you know, you flip the pages as you move from state to state to follow the highway. But really, the concept now of an atlas is it's more, so you take that and then you move to GPS, but now it's more like Waze, the software that not only tells you how to get from point A to point B, but tells you what's the fastest route to get there. So we're talking about dynamic, living, three-dimensional tissue atlases that represent data in multiple different domains. And this network, this Kidney Precision Medicine Project, which is now about 3.5 years off the ground, is a large study and involves 41 different academic medical centers around the US. And we were fortunate enough to compete and become the central hub for this whole network. So we're the data coordinating center, the data visualization center, and the administrative core for this large project that has recruitment sites around the country where people are enrolled and undergo kidney biopsies and longitudinal follow-up and biosample collection. So-called tissue interrogation sites that take those kidney biopsies and do every kind of omics technologies with spatial resolution. And then we have a mechanism of funding new technologies that we bring into the consortium over time. So this is well off the ground now, and we have, at last count, I think 54 biopsies from people with AKI or CKD and people who are undergoing longitudinal follow-up around the country. We're taking these data and doing what's sometimes called multidimensional stacking. So genomic data, epigenomic data, transcripts, microRNAs, proteome, metabolome, collecting all this data and then using a variety of computational tools to integrate these data together. Developing a controlled vocabulary for everything in the kidney and new ontologies, basically to understand all the different cells in the kidney, find the right markers for all these cells in the kidneys, and then collect all this data, and then organize the clinical data, the molecular data, and the pathological data together. So this is an effort that's well underway right now, and we just put out actually last week, there are now 1,500 different files of data that we've put into the public domain on the website as a resource for the entire community to use, with predominantly transcriptomic data and pathological data and clinical data. But the idea is for this to be analogous to the Cancer Genome Atlas, TCGA, which similarly the data was put into the public domain and has been used in over 10,000 different publications. So the strategy here is to make all these data available to all, to clinicians, to investigators, and hopefully transform outcomes for people with AKI or CKD over time. This Kidney Precision Medicine Project, there are a lot of precision medicine initiatives. This one is pretty unique because we really involve patients at the center. Patients are equitable partners. They are on every committee with voting rights, including the steering committee, and we're addressing the big public health problems in kidney diseases. So I'll sum up that we've developed tools, Kidney on a Chip, kidney organoids, and the Kidney Precision Medicine Project. We think of these as all being synergistic and usable to understand pharmacology, toxicology, disease modeling, create new biomarkers, target engagement markers, deep learning, all technologies that interchangeably can use these tools to develop new therapeutics for kidney diseases. And the last slide, this has always been one of my favorite slides. This was the first ever medical text translated as superior doctors prevent the disease, mediocre doctors treat the disease before evident, inferior doctors treat the full-blown disease. And I've always liked to think that nephrologists were superior doctors, but I'm afraid we are, by this categorization, inferior doctors treating full-blown disease. But I'm hopeful with all the technologies that are being developed that we can become mediocre doctors and treat the disease before evident. So thanks very much for your attention. These are all the people who actually do all the work, and then I go around and give talks about it. But we have a great network of investigators for this consortium, all at UW, all young investigators, all thriving and doing very, very well. So I'll stop sharing my screen. And I ran a little over time-wise, but I see one question here. Oh, it's not a question. What about urate from Gordon and Starkbaum? Yeah, urate is, of course, interesting. We have not particularly studied urate nephropathy. I will say, I didn't talk about it, but if I go back, or, one of our funding sources is CASIS, Center for Advancement of Science in Space. Actually, we probably got more publicity from this grant than any others, but we were funded to put our kidney on a chip onto the International Space Station. And the idea there was because the astronauts get a lot of kidney stones, mostly from bone resorption, to see if we could come up with preventative therapy. So we have created an oxalate nephropathy on a chip model, and we could do a urate nephropathy model. We haven't done that yet. And I see a second question, have you introduced immune cells into your organoids? Great question. It's the next frontier because clearly immunity, adaptive immunity, and inflammation are critical to most kidney diseases. And that's where Keith and Christian Lude and we recently got funded to create a lupus nephritis model where we hope to, and it's just getting off the ground. I didn't put any data in because the data is very early, looks promising. But we are going to try to incorporate immune cells. We know that we can incorporate almost any cell type into the various platforms that we have because we've done that in some of the other models. We created a tumor angiogenesis renal cell carcinoma model on a chip that's currently funded. And there we incorporated cancer cells, really remarkable, into the interstitium of a chip and showed that they were angiogenic in ways that proximal tubular cells are not. So we can incorporate other cells into our chips. Organoids are a little trickier because the cocktail that you need to differentiate pluripotent stem cells into intermediate mesoderm, which is where kidney comes from, is going to be different than the immune system. So it's a little trickier for organoids, but certainly for the organs on chips, we can introduce immune cells. And I'll see if, some more questions here. Nope, same ones.
Okay, well, Jonathan, yeah, we have reached 10 o'clock and it's a truly fabulous lecture. I'm sure a lot of people might want to get to you by email or call. A lot to think about and tremendous opportunities, I think, for, like you said, putting together some immune cells, kidney cells, and see what happens. So thanks again, and great to see you.
Great, thanks a lot. Everybody have a good day.
Thank you, Dr. Himmelfarb.